So welcome to Programmatic Access to UniProt using Python. I'm Emily. I'm a curator at UniProt. I help put the biological data in the database. And Dan is with me today to talk about the Python side of things, and he's one of our front-end developers here at EBI in Cambridge. So today we're going to talk about UniProt, and we're going to talk about how you guys access it programmatically. So our mission is to support biomedical research and provide a rich source of always freely available, stable, comprehensive, and as much as possible, accurately annotated protein sequence and function information in the database. We base this on primary sequences, so a canonical sequence, protein sequence. We also have information on isoforms and disease variants. We primarily look at physiological function of a protein, so this is in its wild type state, in a normal cell state, and that includes information such as subcellular location, pathways, the reactions it's involved in, its interactions, and potential involvement in disease. We also include structural information, including topology, experimental evidence of 3D structures, as well as AlphaFold information. And we also have a data analysis tools such as BLAST and multiple sequence alignment. So our main aims for today in this webinar is to help you understand how to access UniProt data and how to pick the best workflow for your analysis, to help familiarize yourself with UniProt and the services that we provide, to know where to find help and documentation if you get stuck with your programmatic access, and to introduce Python and how you retrieve protein entries on the database, including specific fields and filters within your results for annotation type and protein characteristics of interest. If you're not familiar with the database, we do have a recorded webinar online called UniProt, an Introduction for Students, that will give you a basic kind of breakdown of our web services. Over to you, Dan.
Thank you. Okay, so there's different ways to get access to UniProt data. The first one is through FTP, and this is populated with some big files at every release. And these will need some post-processing, some parsing. And there's a little listing of the directory there, and these are split by SwissProt, which is the manually curated entries, and then also we have Tremble, which is the automatically annotated entries. And then another means of accessing it is through the REST API, which is what we're going to be talking about today, and that's for medium-sized downloads. And this is customizable, particularly through the tab separated values that we have. So it's like a table, and you can select different fields within this, and you can use this for a one-off, or you can have it as part of a pipeline and a script and that sort of thing. And then you can also access the data through the website itself. And from there, you can have small one-off downloads, which are, again, customizable through the fields and also the different formats that are available. And something relatively recent is the website file generation. So when you have more than 10 million entries, you can submit a job, and our server will generate the file, and then you can then download that later. And this is also customizable. Okay, so we have quite a few endpoints. The most popular one would be UniProt KB, which is the knowledge base, and it has all of the annotations. Yes, all the annotations that are both manually and automatically annotated. And we also have UniRef, which is clusters of proteins based on sequence similarity. And we have UniPark, which is a sequence archive, and we have a database for proteomes. And we also have some tools. So we have ID mapping, which is a tool that UniProt itself runs. And then we provide access to the BLAST and Align tools that are provided by EBI. And then the peptide search, which is provided by the PIR service in the USA. And so those last three are actually run elsewhere, but we do enrich the results of these things with our own data. And so aside from these main databases and tools, we also have supporting data. We have taxonomy, keywords, citations, human diseases, cross-reference databases, subcellular locations, and then these smaller miscellaneous things. So Unival and Arba are the things that are used to create annotations for Tremble. and then Unisave, which has all the various entry versions over time. But today we will be focusing on UniProt KB. And we have a Colab notebook that we will work through. And I will give you a link to this at the end, but you can also access the API through other means. It could be a local Jupyter notebook or an interactive interpreter with Python or IPython through script, or in any other language, for example, R and Bash. It's because our API is REST and it's using HTTP. So it's quite accessible to many different scripting languages. And speaking of HTTP, it's good to keep in mind the status codes. So if you have a status code for a request that's 200, that means everything is good. And if you have something that's 400, for example, 404 is quite a common thing you might see in other websites. It means it's not found. And it's usually because you've got an error on your side. So you've got the wrong URL or some typo in the query or parameter, something like that. And then 500s is something that we've done wrong on our side. And so we should see this in our logs, but if you keep seeing it, do get in touch with us and we'll try to resolve it. And so with HTTP, again, we have these response headers, and these are kind of like the metadata of the request. And so particularly to the search that we'll be coming to in the demo, we have this header called X total results. And so that is the total number of entries found for that search. And it's not necessarily what you have in the payload. We'll come to that in a second to be clearer then. And then we also have the X UniProt release and release date headers. And this is indicating which current release of UniProt you're accessing. So we have a release every 6 to 8 weeks. And this will just tell you where you currently are. And it's not particular to UniProt KB. It's for the whole of the UniProt. And then we have a link header, and this is for pagination, which it ties into the external results. That will be clear when I show the demo. And then finally, we have content type, which is specifying what actual kind of file format you've requested. Yeah, let's look at these now in the demo. Okay, so here's the Colab, and first we can do some basic setup. We're going to use the requests library, which is very useful. It just provides lots of convenience methods to send the request itself. And we have a few constants here. This is this variable's website API. So this is our REST API URL. And then we'll also be looking at the proteins API, which is a little bit different. It includes UniProt data, and also it's kind of a data mashup. It has variation data from 1,000 genomes, exact ClinVar, and these sorts of things, and also proteomes and human proteome project data, antigen sequences, all sorts of stuff. So it's a nice resource to look at as well. And so this is just a basic helper function for when we submit the request. Make sure everything's okay. If it's not, then it'll raise an error for that. So this is the first basic search we'll make. So we've got the namespace here of UniProt KB, and we're going to search, and then the query parameter here is saying we just want to look for everything with a star there. And so by default, everything returned will be in JSON. We'll get to the specification of formats later on. And then what we're going to show as well is the concept of pagination. So this search returned 25 results. And let's look at the headers now for that. So there's actually 248 million results, because that's returning all of UniProt KB. But we're only returning 25 at a time, because if you returned all these at once, it would be huge. So we provide pages at a time of the results, and you then make success requests. And the next request is provided in the header, and that's the link, as you can see here. So this cursor will get this kind of an ID that tells you what the next page results, where to get it. And then the release and the release date are also listed here. So we're in the 2024-05 release. Okay. So to make a query, I just did the star search, which isn't that interesting, but you can do two types of queries. You can do a free text, which is where you just, whatever, parking, for example, we have in the screenshot here. And so that will then look over all the index fields we have in the database. And then you can also use a more specific thing, which is the Solr format, or the Lucene query language. And this is a way to specify the fields and the values, and you can have logical conjunctions to join all these things together. So in this screenshot here, we're using the advanced search to construct a more complicated query. We're going to have a free text search of Parkin, and then we're going to specify that this wants to be in the Homo sapiens taxonomy. So let's have a look at that, the demo. Okay, so here's just as we had before. It's just join these together in and we're now going to iterate through the pages as they're provided. So let's run this. Okay, so we're adding together the set of results as we go. So initial page is 25 results, and we know there's 144 thanks to the X total results header. And we're just going to keep going until there's no more links available. So at this point, we have everything here, which is quite nice because you can kind of get a progress. So you can work on each batch as you receive it as well. Now there is another way of accessing it, and that is using the stream endpoint, which is in some ways simpler because you don't have to have the logic of this pagination, this while loop. But it has the issue of being a little bit harder to follow progress, and because it's just one relatively big download, if something goes wrong, then it will just fail and you have to resume. You have to start all the way from the beginning, whereas in this setup here, you could have some logic to resume from the last successful page. And it is not sorted by the search score. So when you do a search, the results will be sorted by what our backend considers to be most relevant to that search. So when you use the pagination, it will be sorted. And when you use the stream, it will be in a random order. And then finally, the stream is limited to 10 million entries. So let's just go ahead and run that. Yeah, so it's just returned everything at once, 144. Okay, and we can also specify different formats. There is the full list available from this link here. I'll run through a few here. So let's first of all show the JSON. It's a very long string, so there's quite a lot of information in this. You have the sequences, sequence updates, annotation scores. There's a whole bunch of stuff in here. And the good thing about JSON is that it is useful for working within Python because you can just convert it to a dictionary, and then you can traverse it and pull out what you need as you go along. So we also have the XML format, and in order to specify this, so in this line I didn't specify anything, and so for that it will use the default JSON. But here I'm going to say format equals XML, so let's run that. And we get an XML file, which you can also traverse with Python's XML library. And you can also specify a different way of getting an XML, and that's using the accept headers, which we looked at in the slides. So if you set this application XML, it will return exactly the same thing. I won't bother running it because it's just the same thing as before. And then another file format is the list, which will just return the accessions that are returned from that search. And then we have FASTA, which is the sequences with the FASTA header. So this is, we're using the search, so there'll be 25 of these. And then likewise, this is a very popular format, the TSV, which is tab separated values. So this is like a table of results here. And this is the default fields that were returned. And you can also customize these fields by using the fields parameter here. So rather than having the default, I want ID accession length, and then I want catalytic activity. Running this returns this. So the columns have changed to respect the fields here. Okay, we'll head back now to the retrieve and entry slide.
So let's have a real world example of how you'd use Python and compare it to how you'd use on the website. So if you have an entry of interest, let's say your mass spec gave you a UniProt accession ID and you wanted to see what that was, you could put that accession ID in the search field on the home page of UniProt, and it would navigate straight to a protein entry. So in this case, we've stuck with parking for the human entry. The header will show you the full protein name and its gene name, its status, which is a review SwissProt entry. It's in humans. It's got 465 amino acids, and it's got the top protein existence and annotation score. And then what you also have at the top of your entry, protein entry form, is function, if the protein's got a function. Then you can scroll down or use the tabs on the left to navigate the full protein entry page. So Dan's going to show you how to pull this information back in Python.
Okay, yeah, so we will do this back in the Colab, and what we're going to do is we're going to try to pull out the catalytic activity, which corresponds to this little screenshot here, which is actually from the entry page. So, first of all, you can just say, I want everything. And so there's no format specified, so this would be JSON. Let's go ahead and have a look at that. And you get a whole bunch of stuff, a whole bunch of data. You have database cross-references, you have the sequences, you have features. I can get to the top. Yeah, it's a lot.
This is a big entry. We picked a big entry as an example.
Yeah, and citations, everything you can see on the entry page on the website, the data, underlying data will be in this JSON object. So let's be more specific now, and we want to have the specific field of CC catalytic activity, and we'll run this. And now it's filtered it on that comment type, and it's pulled this out. Yes, so all this information that you see here is being used to populate the actual information on the website.
So I'm going to talk a little bit now about how we annotate at UniProt, because there's no point looking for a set of data if you don't know where to find it and how it's put into the database. So we make all our data as traceable as possible, whether that's through cross-reference imports from other databases or manual annotation by curators. So these are just some examples on this page of how we evidence tag fields and data. So in the names and taxonomy section, we have obviously the recommended name for the protein and parking as an EC number. So next to the EC number, you can see here highlighted with the first red arrow that we have 2 publications associated with this EC number. That means those 2 publications have evidence to support Parkin's role in that EC, in that reaction. If you click that little grey evidence tag, you get this extended grey kind of field box, and within there you get the information that says this is a manual annotation based on experimental evidence. So that means a curator, such as myself, has put this data in from the paper. You have the paper title and its author list, and links to PubMed and Europe PMC. And a little bit further down, if you were to scroll down in that box, it tells you what other UniProt entries this paper is also cited in. Further down, and the second red arrow in this example is an imported evidence tag. So this is for an alternate name. This tells you that it's a manual annotation. A curator's touched this, but it's imported from a database. So in this case, it's HGNC that have given this alternate name for parking. And if you click that HGNC link, it takes you straight to the source page at HGNC. And then on the right here, we have subcell localization examples. So this protein is expressed in the nucleus with a supporting publication in those tabs. And then further down, we've got mitochondrial outer membrane with a bisimilarity tag. Now, this is when we know these proteins are conserved between orthologs. That data in the paper is maybe shown in an ortholog entry. So that the data in the paper will be, as an example, done in mouse, whether it be in a mouse model or a mouse cell line. So that information is added into the mouse entry. And because that protein is conserved between mouse and human, the information has been propagated over to the human entry. The bisimilarity tag will include a link to the mouse entry or the original entry where that evidence is from. So that's how we, on the website, tag where the experimental and cross-referenced information has come from. Next slide. Thank you. And we do this in the database using echo codes, also known as evidence codes. So the tags are literally website translations of these echo codes. These are just a few commonly used examples that we use on a day-to-day basis, but there are a full list at that URL at the bottom of the page. I'm going to touch on these a little bit so you can understand that how we curate the data is different depending on the evidence available. So echo code 269 is experimental evidence, which means a curator such as myself has looked at that paper, said, yes, there's experimental data to support that statement, and we've added that echo code and tagged that PubMed ID into at the end of that sentence or at the end of that field. Echo code 250 is a sequence similarity, which I've just mentioned. So if the data is in mouse, cell line, or a model, that will be added with an experimental evidence code, so 269 in the mouse entry, but in the conserved ortholog in the human, it will be a 250 code, which will tell you this data is in the mouse. We have echo code 305, which is curator inferred data. So this is when a curator has read a paper and the author says this protein does X, but there isn't quite enough experimental data to definitively say that and make that statement. But as is normal, the curator would have a feel for the field, the signaling pathway, or the protein family, and we'll be able to say that that makes sense or that fits within the field and experimental knowledge that is currently out there. So we add a curator tag, or in this case, in the examples of the evidence tags on the right, it would say curated. And then the final example I've given here is echo code 312, which is imported evidence. So this is what we had for HGNC in the earlier example, where that data has been brought in from an external database, and the link should be there to navigate to that external database. Next slide. So we mainly curate our protein entries are based on canonical sequences of proteins, but we do curate information specifically to isoforms as well. In the database, we have, if you're in a section like this, which is subcell localization, the first comments will be generic to the canonical sequence. So this is where the authors and the experimental data do not differentiate between isoforms. They say that protein is in the nucleus. Brilliant. So we tag it in the nucleus. Great. The isoform data has to be from an experimental derived procedure that they can specifically say it is that isoform, not another isoform, not the canonical. So these antibodies must be specific to isoform 1 or these primers specific to isoform 2. And in that case, we would curate them such as this. So it will be under the isoform 1 header that's specific to nucleus or endoplasmic reticulum. However, isoform 2 is specific or has been seen in the nucleus and cytoplasm. And they would also be tagged with echo codes and evidence tags as well. So in Python, to specify this and to bring forward the isoform information, you would have the accession ID in your search query, and then you would say include isoforms equals true. So that will feed you back the isoform information. And in this query, you also get the accession. You're asking for the accession ID, the function, the subcell localization, the PTM, etc., etc. And then in your results, the first function comment you've got with the accession ID is for the canonical sequence. So you notice it's accession number with no dash after it. So that will be the generic function that isn't specific to an isoform. And then the second function comment here is an accession ID -2. So that's specific to the -2 isoform. That evidence is specific to that particular sequence and has been shown experimentally. Next slide. So you can pull all that back from Python, and I'm going to move on to our next live example now, which is how to find natural variants on a protein entry page and then programmatically. So when I say natural variant, I mean seen in a patient, seen in the real world. We had someone walk through our door, we sequenced their genome, that variant is in there. I don't mean I took a mouse, I genomically scrambled it, and that is a variant. We call that a mutant or a model mutant. It doesn't include cell line data, so nothing done in the lab. This is all a human walk through the door and their variants are in their genomic sequence. So these variants are obviously in UniProt displayed for their protein sequence. And as you navigate to a protein entry page and you go down the page, you have a disease and variants section. We will, if they're involved, that particular protein is involved in a disease, we will have the description, which is imported from OMIM. And you can see here that Parkinson's disease, interestingly enough, Parkinson's involved in Parkinson's disease. And we have 4 publications to support the involvement in that disease. Below the description, we have a features graphic and table. So from left to right, we have the amino acid string sequence, and annotated on that with the black bars are the variants that are available that we've got evidence for. You can click on the graphic or you can navigate down the table, and it will show you the variant IDs, its involvement in the diseases, if there's DB SNP annotations, and if there's any further characterization, i.e. this variant, when we examine it, does X, Y, and Z. That will also be curated there if it's done in a human model. Also on the entry page, we have our variant viewer. The tab is at the top of the page. This again is a graphical view of the amino acid string, N-terminus to C-terminus, left to right. You can scroll and zoom, and each dot stands for an amino acid variant found on the sequence. You can filter via provenance and consequence, provenance being source. So you have UniProt curated variants here, as well as things brought in from cross-references such as ClinVar, and your consequence, i.e. does it cause the disease? Is it benign? Do we just not know? So you can filter the information there. And Danny's going to show you how to do this with Python.
Okay, back in the collab. So we're going to try to access variants in 2 ways. One way is through the REST, UniProt KB REST API. And then we'll also look at the proteins API, the variation endpoint. So we'll run the first one here. And this gives us 40 variants, and we're just looking at one example. And so it tells you the type and the location in the protein coordinates of where it occurs, and the feature cross-references, so from the database, in this case, DB SNP and the RSID, and the echo codes, as Emily just spoke about, and the feature ID. And then lastly here we have the original sequence, so I guess wild type, and then also the alternative sequence, which is what is changed to. And then we're going to run this, which we'll use the proteins API. And so before we had 40, and here we have nearly 900. Nearly 900 variants. So there's the proteins APIs does pull in a lot of variation, and it maps it all. So it's coming from ClinVar, 1,000 genomes, EXAC, Nomad. So going through this here, we have the alternative sequence, and we're at the very start of the protein sequence here. So it's just at position 1. And then we have all the cross-references, so ClinVar and DB SNP, and we have the links to the corresponding entries in ClinVar and DB SNP. And then you have the cytogenetic band to show you the chromosomal region and the locations here, and the consequence type is missense, and then the wild type, and then what's mutated to. And we have population frequencies. So this is the minor low frequency, and that's coming from ClinVar. And we have the clinical significances, again, coming from ClinVar, and it's considered to be pathogenic. And then we have disease associations. So this is autosomal recessive juvenile Parkinson disease 2. And it has the OMIM link and evidences with the eco codes, which Emily just spoke about. And then let's return back to the presentation where we'll talk about retrieving and comparing orthologs.
So we're going to talk about something a bit different now. So obviously when you're doing experiments, you want to know if these proteins are conserved between different model organisms, and whether it's useful to study in a different species. So this will be the only time you will ever do a generic search on UniProt for a name. The rest of the time you should be using the advanced search. But I've put parking in the search bar and hit search, and the results come up. You get quite a few because this is searching the whole database, all the entries, anywhere in the entry for parking. So whether it's mentioned in a function comment or miscellaneous comment, that entry is going to be there. I'm obviously only interested in the protein parking. So I've manually selected here in this results set the Drosophila, the human, the mouse, and the rat. And I've done that by clicking the little tick box on the far left in the results section. And then I've clicked add to basket. And you can add to basket to do tool searches or analysis, or just to save things for later if you've run out of time. So next slide, Dan. When it's in your basket, and your basket is in the top right of the web page, you can open your basket to the full size and select the proteins that you're interested in analyzing. So again, on the tick box on the left, I've selected all 4 of them, and I've clicked tools and selected align. When you select align, I've not shown it in the screenshots here, but it opens an align web page with an automatically populated form with the canonical sequences for these 4 proteins. I've then given it a name so you don't lose the job and get confused later on, and click go. These jobs occasionally take a couple of minutes to run. Align's generally reasonably quick, and it appears in your tool results bar, which is also the top right on your homepage. Looks like a little workman's box. And when you open that, it will say the job is first queued, then it goes to running, and then it will be completed. So like it's got here, it's completed in blue with a little green dot. And if you click completed, it opens your results page. So this is a default view on the align results page. It's aligned the sequences according to similarity. The darker the purple, the more conserved the sequence. So the deep purple here is conserved amino acids between all 4 orthologs. And then the paler purple is one of the orthologs might be missing, might not agree with the other 3 orthologs like we've got here for the start of the mammalian sequences. And then the dashed lines are where you've not got a sequence that aligns to another sequence. So as you can see, for the Drosophila sequence, we've got an extended N-terminus for parking that doesn't match any from the orthologs. And the mammalian orthologs are missing the section further down that the Drosophila includes. So Dan is going to show you how to do this in Python, and he's going to use the exact same accession IDs.
Yep. Yes, so I use, as Emily just said, the same accessions. And first of all, we need to grab the FASTA, so we need to get the sequences for that. And we use the accessions endpoint and specify what FASTA format. Let's run that. And so here we have the FASTA sequences. And then we're going to use that and run the job here with this post request at this endpoint. So this is the EBI's cluster O algorithm. And this dictionary here will specify the parameters of the job. And you can put your email address in there. I've never done that. I've always just polled the job, which we'll see in a second. And then iterations, just the number of HMM iterations that the cluster O will run. And the output format, we want to have cluster num. And then the order of the results can either be in the way that they are aligned, or you can have it returned in the way that you've sent it to the cluster O. And then finally, we're just saying, here's our sequences. So let's run this now. And what will happen is when we make that request, we will get a job ID. And you can then check to see how it's doing by polling the status. So we would keep running that until it says finished. That usually quite quick. So that's done now. And let's have a look at the results now. So it's a similar view that we saw that Emily just talked about. And rather than having the shades of colors, we're using these text characters to represent the degrees of conservation. In the previous screenshot, there were shades of purple. Here we have the asterisk fully conserved, and then the colon for conservation between groups of strongly similar properties, and then it just decreases down weakly, and finally non-conserved residues. Right, we will now return to talk about retrieving IDs through the ID mapping service.
So this is a good example of another one of our tools where if you've done an analysis on say a mass spec or whatever in the lab, and it's given you a set of IDs, and then you want to do something further down your analysis pipeline, but that next bit of software doesn't take those IDs, it takes a different ID. So a good example is your first experiment spits out gene names, and your next piece of pipeline accepts UniProt accession IDs or ensemble IDs, as an example. With the ID mapping service, we allow you to map between these different IDs, with the only parameter being you must pass through UniProt IDs at some point. So if you're going from, as an example, gene names to RefSec nucleotide IDs, you have to go gene names to UniProt IDs and then UniProt to RefSec IDs. So bear that in mind. But you can find the ID mapping service in the top left of the web page or on our homepage. And that gives you a query page like on the left here. You can load a text file in full of IDs, or you can copy and paste your IDs into the box. You then tell it where you want to go from and to. So in this case, I've said we're going to go from UniProt IDs. We have a list of 4 here to RefSec nucleotides. We have an extensive list of IDs that you can select. So please go and explore that if this is something you're interested in. Then name your job. A word of warning, if you are going for gene names, especially from gene names, there is an option to specify your tax ID. Please specify your tax ID. If you put gene names in, you're going to pull back IDs for every species we have in UniProt. That's going to be a very long list. Please specify your taxon ID. Then name your job, something better than that example there, so you can find it again. Run your job, and then when it's finished running, it will be in the tool ID tools results box that I showed you in the previous slide. And when you open your results, you'll have a table like this on the bottom right. So you'll see your from IDs and then what you've converted it to. And you can download that as a text file or as a simple list file. And you can just you can mess with the settings there, so you can just download the IDs that you've changed them to, and you can take that text file and stick it straight into the next section of your analysis pipeline. So Dan's going to show you how to do this programmatically.
Yep. So also there's a question from Bernice about can you start the search for list of ensemble gene IDs? So this I think would be a good application of doing that. So you can search for single gene ID, but you could also have a list using this particular service, the ID mapping service. So let's have a look how to do this programmatically. And it's a similar workflow as what we had before, of where you make a post request and you populate this dictionary with the parameters of the job. So we're going from UniProt KB accessions. I'm going to go to RefSeq nucleotide, and we'll just use this manually selected list of accessions here. And then we'll submit the job, and we'll get a job ID. And it's already done. So we've been redirected to the results. And so you can see the UniProt KB accession and what it has mapped to for the RefSeq nucleotide. Yeah. And so here is the link to the collab I've been running. You're welcome to grab it and edit it and run it as you like. There's also other code snippets in there. There's some BLAST, some visualizations, and retrieving PTM exchange data. You can also use this QR code to access it here.
So I'd like to thank our funders. We're funded by the NIH and EMBL, Open Targets and CIRI. UniProt is a consortium here at the EBI, as well as PIR in America and CIV in Switzerland. And the next slide shows our whole team. We're all around the world with curation and development teams at all three institutes. It's quite a big group of us, actually. Next slide. So help and training for UniProt. This is UniProt specific training. On our homepage, we have a help and training section that has just been revamped. So we have, it will tell you what up and coming webinars we've got once they're live on our training websites. We have tutorials and videos, which are all pre-recorded and you can watch at your leisure. We have online courses that are interactive. We have training that you can attend at EBI, PIR and CIV that are UniProt specific. And we're also on social media. So we're on Facebook as well as X. Please follow us for updates about the website. The webinar that I suggested, UniProt for Students, is on our tutorial and videos, and I believe Anna's put it in the chat box as well. If you want to contact us or you need help, there's always a help tab on the right-hand side while you're navigating the website. You can search in there for any relevant information you're struggling with, or you can contact us using the Contact Us form. That will get sent to a help desk and sent to the most relevant person to help you with your query. You can do this if you spot an issue with a database, like something not running or an error, or you can contact us if you're stuck with your code as well. We will help if you're really struggling trying to access our data or you're looking for something specific. The best bit of advice I can give you if you're looking for something specific in your protein entry pages, programmatically, is to get used to the website and get a feel for where your data is. Go practice, basically. We have several up and coming webinars, and I realize these are a little bit further in the future than we normally are, but our next one is in December, when Michelle will be looking at functional annotation and I will curate in UniProt. In the new year, we have an automatic annotation machine learning webinar and how we're using that in UniProt. In February, we do a webinar on our tools. So this covers BLAST, Align, and ID mapping. And then you're back with me in March doing a UniProt for proteomics scientists, and we do touch on API access as well. We've got new stuff in the website all the time, but at the moment our newest stuff is Alpha Missense track on the feature viewer. We have complex portal visualization has been integrated in the interaction section of our protein entries. And coming soon is our next data release at the end of November, which is release 2024-06. We have GoCam visualizations being integrated soon. And we've got a new Swagger REST API documentation currently in production. I think I covered everything. So we're going to go to questions. I think we've got some good ones, Dan. I'm going to take this one, why I see it. So can manual annotation be rectified based on further evidence? Yes. So what we do is if I see an entry that hasn't been updated in a while, I will take that, look at the literature and update the information that's in there. If a user writes to us and said, hey, you're missing this data, or this data is wrong, or its canonical sequence has changed, we would look at that too, yes. We try and be proactive as much as possible. But, if you write to us and you spot a problem, yes, that can all be changed as long as there's evidence out there. And that will be evidence we accept, specifically endogenous function and physiological function. So we wouldn't necessarily take it if it was in disease cell lines, but we would if it was in a tissue, as an example.
Oh,
right.
There's a question from Alejandra. How can we select the specific version of the database to do the search? So you can't necessarily select the version to search. It'll always be the latest. In terms of historical releases, that's available from the FTP. And you can also get the entry version for every accession we have in UniProt-EBI. So you could determine when it was introduced to UniProt-EBI or when it was moved or merged or removed as well. So there is some history for each entry available, but for the overall search, you only get the latest version.
That's worth stating if you're downloading our proteomes and doing, mass spectrometry searching or big analysis. Always, when you publish, always state what version you use, because this obviously changes between releases, and that will change your results. What is the main sources for the orthologs? So in my example, I put a protein name in, and I made the assumption that the proteins in the orthologs would all be named the same thing. I know that's the case for parking. I cheated. Okay. So normally you would use a line to look at orthology and sequence conservation, which is what we did there. We don't actually have a link for you to pull all orthologs back. So you still have to look at OrthoNet or other sources to see where orthologs are. It is something we're looking into. But essentially, when we curate ourselves, if I'm looking at the mouse entry and I'm adding data to the mouse entry, I would align all the other entries that we have in the reviewed side of the website and say, how far can I copy this data? Is this, if I'm curating an active site, is this conserved across all mammals? Is it conserved into Drosophila? Is it conserved into fish? We will make that judgment based on alignment data as we curate ourselves.
Okay, I'll answer Mohammed's question, which is there a size limit in terms of bytes for the result set? So there isn't a limit in that respect. The limit is more about the number of entries. So for the stream, you cannot stream more than 10 million at a time. For pagination, you can run through all the pages to get all the results set. So it's not limited in that respect. And if you don't want to do that pagination business and you have a lot to download, a lot of entries, best way to do it would be to use the file generation that's available from the website. And that's available for more than 10 million entries. But we don't necessarily limit on the bytes.
Dan, do you want to take another one? I'm trying to answer a question by looking at the website. Bear with me.
Yep. Is there any performances differences between the search made with a field selection and without? This is asked by Susie. There is, in that we have a default set of fields, and if you perform a query that's often asked, you will get a cached result. So it's a means of speeding up the performance of the site. So it would have already computed this and returned to you quite quickly. So if you ask for something which isn't in the cache, then it'll be a little bit slower because we'll have to actually go and perform the search. And then also, if you have a lot of fields, it would just take a bit more time to produce the result set because we have to go to the database and fetch each field for each entry. So it'd be a little bit slower if you have a lot of fields. It would be proportional to the number of fields that you add.
So I want to answer the question about proteomics. Using a reference proteome, and they've said the human one, would it only include canonical sequences? What should you use if you're looking for all specific isoforms? Or fusion proteins. We don't have fusion proteins in the database. If you want to do, if you're doing a bottom-up experiment and you want to map to a fusion protein, you have to manually add that fastest fusion protein sequence into your search file. Copy paste it. However, if you want to download the canonical sequence in isoforms, can I steal screen down?
Yeah.
I don't know how to do this. Hang on a minute. I did. Share screen. Share. Because this might just be easier for me. Oh, I've got stuff everywhere. Okay, so I'm on the human proteomes page, which is what our participant wanted. You can dead simply download one protein sequence per gene. That is your canonical sequences only. Okay. If you want to download For the isoforms, you go to Components, Download, Include Reviewed SwissProt Isoforms. You can only do faster formats here, okay? And then you select, do you only want our canonical sequences or also our unreviewed sequences? So what this is asking is all our reviewed sequences we've got canonical forms, okay? Some of them have also got isoforms attached to them. So that is saying, do you want the canonical proteins here? And then I've ticked this box and said, all those reviewed entries that have got reviewed isoforms, that is what I want. If you also want to include unreviewed entries, and in an unreviewed Tremble entry, the isoform has its own entry ID. Okay, so when we bring these isoforms into a canonical sequence, we merge them. So you will see they will have the same accession ID in a -2, -3, like Dan showed in the Python example we gave earlier. When you're looking at the unreviewed side of the website, they will have a completely unique ID. So if you want a true view of absolutely everything we have, you want to go include reviewed isoforms, as well as download all reviewed and unreviewed Tremble entries. That file's quite big. If you only want the isoforms that we've got evidence and data to suggest they exist and they've been reviewed, and you're not interested in the automatically annotated Tremble side, you change your top selection to download only reviewed. If you've got any fusion proteins, any variants and mutants that you think you're interested in your proteomics data set, and the sequence is the protein sequence is different, and you know that peptide will be unique to that protein, you have to manually add that FASTA sequence into your file, just as you would enolase, as an example, when you spike in. Stop sharing now. That was nice. That was a technical question. I like that one. Got it?
Okay, so there's a question from Bernice again. How do you specify the taxa in the online tools programmatically? So I'll show you how to do that on the website. I've just run it actually, so I'm going to hit the resubmit button and then it will show us where this appears. So I'm going from GNAME to UniProt KB, and then within this, there is a restrict by organism, and it'll autocomplete on which you select. And then also, in order to do this programmatically, you would just specify the tax ID in the request itself.
Please do that. If there is an option on the UniProt website, it makes your life so much easier and your searches so much quicker.
Did you want to take the question from Scott, Emily, about can one take lists of human proteins and create a list for each of its splice positions based on GRCH38?
I was hoping you were going to answer this one. So I think we can pull this out using the genomic coordinates service now, can't we?
Yes, that's.
And I think this is easier programmatically. You can do it on the website. So if you go, just click a protein entry on the website. See, Dan's got one ready here. So you've got the genomic coordinates tab, and it will give you all the coordinates of all the isoforms. And then further down, it gives you, should give you a set of exons and splice sites and what isoforms it refers to. And then you can do that programmatically using the coordinates service and the proteins API, I believe. Get me. I don't even program and I know this.
Okay, there's a question from Shonak, and that is UniProt also gives external links like cake pathways or gene expression or family, protein interaction details. How can the data programmatically be integrated from those external links? So we have the list of cross-references, and we have some metadata about the cross-reference entry, and we also have the IDs. So we have that available. However, if you need more, you would probably have to make a request to the corresponding cross-reference to obtain that information. Yeah, I think you have to assess for each of these to determine what we have in the features available and the cross references available. Yeah, I think that's anything to add to that, Emily?
Yeah, so each of these, so the example is interesting because protein-protein interaction details are going to be in a separate section of the entry to KEGG pathways, right? So you want to already know where that data is on a web page for you to be able to pull that down programmatically. So if you go in interaction down on the far left, so if you scroll down, APP's got a lot of nice manual data. So all these binary interactions are programmatically available. If you scroll down again, on the main page, we have our complex viewer that we've just imported. And if you keep going down, you have more cross-references. So they're going to be in the interactions side of the programmatic pull down. You're going to have to search for them specifically, just like we did with variants. Whereas your KEGG pathway results are going to be in just Control F pathway for me, Dan. Oh, that's unlucky. Go again. Organism specific data. Keep going. You've got like 10. There we go. So now we've got Go terms. There we go. And we've got keywords. So these are all in different sections of the entry. So you're going to want to have to filter through these sections to look for your information. This is what I said about get familiar where the data is on the website and where you can pull these out from. So you see you've got all the Reactome links here, and I bet further down you've got, Go and KEGG pathways as well. Family groups. But yeah, you can pull all that data back. Oh, and I've got to answer this one, Anna, because this is a bugbear of mine. I know you've just popped up to tell me we're near time, but how will you go about searching all human enzymes in a class such as methyltransferase, and then only include those that act only on proteins and not on DNA? So in the advanced search, you'd say I only want to look at tax ID human, number 1. And then you say and ideally you want the class of enzymes using its EC number. If the enzyme or the pathway hasn't got an EC number, I would say you want to stipulate in function. So you can specifically say in function, methyltransferase will be there if it's in function, and we've got a curated enzyme for that. The last part of the question, then only include those that act only on proteins and not on DNA. I'm not sure there's a function for that. I'm not sure there's a way of doing that. I think you'd have to pull that data out yourself. But what you can do is when you do your tax ID in human and let's say methyltransferase in function, you get a results table. If you go into customized columns, you can say, I just want the protein names in the function column and maybe the interactions, and you can just sift through there. Add stuff to your basket and just hit download, and you can download that as a file. And Dan's not got time to show you that programmatically because that's like a massive, that's like several filters. But please use the customized column section because it means that whatever you download from that page, you can completely customize what information you're getting back from any search you do in UniProt on the website. And that will translate to programmatic access as well.